Greetings and welcome back. In the last vignette, we talked about the cell as a bag of X, where X was a variety of things. It was genes, proteins, alleles, and so on. And all of that was a precursor to this amazing age of DNA sequencing and RNA-seq and methodologies for counting RNAs. So what we're going to do in this high-paced vignette is talk about the cell as a bag of RNA. I already, in the previous vignette, showed you this graph, which came from attempting to scour the literature to try to find out when the expression bag of enzymes had been used in the 50s and 60s, and then in its decline, and then the rise in thinking of the cell as a bag of RNA since the inception of RNA-seq. So like I did in the last vignette, I just want to remind us that there are different ways of going about the experimental measurement of RNA molecules. One of those methodologies shown at the top is RNA-seq. In broad brushstrokes, the idea is that you have a whole series of different mRNAs within a cell. You break open that cell, you amplify through reverse transcription that RNA in order to produce the cDNA, and then that is sequenced, and we have some sort of a count of the RNAs. That hides a lot of sins. And what I mean by that is that this is an incredibly hard experiment. It's demanding. There are many steps in the process that can get in the way of accuracy and precision. But for our purposes today, we're going to assume that we can do it. The next thing that I show you is an example of a fish experiment, fluorescence in situ hybridization. And this is an alternative for counting RNAs. It's an alternative based on fluorescence and microscopy where, as you can see on the left, we order some probes for an RNA of length a couple thousand. You might have as many as 60 probes. Each one's on the order between 20 and 30 nucleotides long, each one of which harbors a fluorophore. And so when all of them hybridize with the mRNA of interest, you have on order 60 probes per mRNA, and so those show up as spots in microscopy. And here too, one has to be careful. It's not trivial. It's not easy. One has to go through hard work in order to measure the RNAs. And I just wanted to say in passing that there are beautiful experiments which are known as spatial transcriptomics. There are a variety of groups that perform these kinds of experiments, and one of the leaders in the field, I would say, is my colleague at Caltech, Long Kai. I just heard a talk from him a few weeks ago, and more or less every time that I see him give a talk, I am essentially amazed by what has transpired in the field more recently. Here, the idea is by multiplexing different colors. Essentially, what you're doing is you're barcoding through color. One can not only count the mRNAs, but also one can discover their spatial localization, which is in itself interesting. And coming back to what I showed you earlier, which is my beloved figure from Perrin. Just to remind us, this is Jean Perrin from his book Atoms. And this table is a statement about confidence in determining a number, in a confidence in measuring something. And what I want to say is, we would like it to be true that all these different methods yield the same answer for the number of RNAs in a cell. And I will leave that to later discussion. So what we're going to do now is we're going to interest ourselves in this giant matrix, which I'm going to call the gene expression matrix. A given row of this matrix is one specific cell, and each column corresponds to a different gene. Gene 1, gene 2, gene 3, gene 4, etc., all the way up to gene M. So the labeling here, GIJ, means the number of mRNAs of species J in cell I. So that's what this matrix is. So another way of saying it that I said in the last time is that I can think of this particular row as a giant 20,000 dimensional vector. What's in that vector is the counts. It's the counts number of mRNAs found in that particular single cell. And really, where we're headed with this is I would like to have a theory of the transcriptome. That's what the objective is here. In order to talk to you about how this works, my plan is to now consider a toy model of this process. And the toy model is one in which we imagine only 3 genes. In other words, we're going to have a 3-gene transcriptome. And the reason I'm showing you this is just to point out that there are examples where a very low-dimensional representation would allow us to define cell type by just looking at the disposition of a few genes. That's really just motivational. In other words, I'm just trying to say the notion of building our transcriptome around a few genes is not that off the wall. So here what I'm showing you, and this is especially directed at students in my course itself because they're doing a homework where they're generating a synthetic transcriptome. And the idea is that I have 3 cell types shown here. And the cell types are defined by the number of copies of the mRNA of gene 1, gene 2, and gene 3. So cell type 1, what we see is that the average value of M1 is 100. The average value of gene 2 is 100. And the average value of M3 is 1. And how we generated this is we exploited the Poisson distribution. So what I mean by that is that we drew from this distribution, P of M, where lambda is equal to the average number of mRNAs. So I could have rewritten this as equal to average M to the M power, e to the minus average M over M factorial. And so these distributions that you see up here, these are the results of sampling from the Poisson distribution with average 100 and so on. So what do I have? I basically have let's say a 3 by 3000 matrix because I have 3 cell types or 3 cells. No, sorry, I did not say that right. I have a 1000 by 3 matrix because I have 1000 cells, but I have only 3 genes. So that's what I mean by the gene expression matrix. And here it is. So, you can go look here at a particular row. So, some particular row right here. And on that row, what we're showing is the level of expression of the three genes of interest. So in this particular case, there's 95 copies of gene 1, 86 copies of gene 2, and 0 copies of gene 3. This is the result of drawing from the Poisson distribution with average 1. Here's a different example. So this guy has 93 copies of gene 1, 111 copies of gene 2, and 1 copy of gene 3. So you can see on the right-hand side, this is the number of copies. So the color is trying to tell us that. But, we actually show you the numbers. Now, what I'm showing you at the bottom is cool because what I'm saying is that, and this is not accidental, we chose the example because we wanted to be able to plot it. So I have a gene 1, a gene 2, and a gene 3 axis. And so what I do is for each cell, I plot a point, which is given by the point of a given point is M1, M2, M3. So those are Cartesian coordinates. And what you can see already is that they cluster into groups. So this is the 3 different cell types. So the one in which I have high, high, low is cell type 1. The one in which I have high, low, high is cell type 2. And the one in which I have low, high, high is cell type 3. So all of that is lurking in there. So if I were to now consider, so this is a new problem. And this is a new problem for the reason that I want to do a two-dimensional plot so we can watch this thing unfold, 2 genes only. So this is not the same as the example above. Now I'm only doing 2 genes. And you can see that the level of expression is either 10 or 35. So, so I have 3 different cell types, which you can see. So this one is, this cell type down here is low value of gene 2 and high value of gene 1. This cell type is low value of gene 1 and high value of gene 2. And this one is high, high. So both on average 35. What I'm showing you here, and this is something that the students are going to do in the homework, is an example of the so-called k-means clustering. And the idea is simply stated, which is we make a guess for the centroid of 3 clusters. And then what we do is we find all the points that are closest to each one of those things. And that's what you see here. And then we calculate the centroid of that residual. In other words, so saying it again, I make a guess. I could do it by eye. And I make the guess that there's 3 clusters, and I center the 3 of them here, here, and there, for example. That's not actually what we did. We did here, there, and there. And then what I do is I calculate all the data points that are closest to each one of those 3 things, and I then find the centroid of those effective clusters. And so that moves the new positions, gives us new positions for our centroids, and we do it again. And what's kind of crazy and surprising is that this method converges. In other words, we actually can cluster our data, although it has a shortcoming in the sense that if you were to tell it that there were 4 cell types, it would find 4. It would cluster 4 cell types. That is something that sophisticated people in the actual field know and know how to avoid and so on. So I'm not criticizing. I'm just saying that's a feature of the most lowbrow version that I've done here. Okay, so continuing with this example, now what I want to do is I want to head towards the subject of principal component analysis. So, in terms of logic, what have I told you thus far? So I have told you, first of all, this idea that I wanted to make a synthetic transcriptome. I defined it for a constitutive promoter. Why do I claim it's constitutive? I claim it's constitutive because it's described by Poisson statistics. And that's something we've talked about in the in-person class. It's something I've talked about in the online YouTube channel, etc. So we produce such a thing. And now, the point here is just to give a flavor to people about the kinds of figures that you might see in a talk or a paper about genomics. We're just trying to demystify it by showing it in some sort of a low-dimensional example with the hope that that sort of makes sense. So, we identified different cell types. Then I made this new problem with 2 genes only. We did clustering, and that's fine. And now we're on to the next thing. And the next thing is the way I'm going to say it, is to try to find the good coordinates. We're back to 2 vignettes ago where I went through all this trouble, which might have seemed gratuitous, about normal modes for a 2-mass 3-spring system. And I'm trying to make the point that no, that was not gratuitous. That was very enlightening. So here what I have is I have 2 cell types. I have cell type 1, which is low, low, and I have cell type 2, which is high, high. And here you see the gene expression matrix. And basically, these are all cell 1. You can see that the color is sort of bluish. So these all generally have about 1 mRNA for gene 1 and 1 mRNA for gene 2. And then these guys, it's more like 50 RNAs on average per cell, drawn from Poisson. Now what we're going to do is we're going to remind ourselves of what we said the other day. What we said the other day was this crucial idea of a statistical summary, which is the covariance matrix, And I remind you that the way that we defined it was x1 minus x1 average, quantity squared and averaged. And then this guy was x1 minus x1 average times x2 minus x2 average. And that is the same thing in the next spot down here. And then finally we have x2 minus average x2 quantity squared. And that matrix is the covariance matrix. So how would I get that? I would visit each data point and I would figure out, what the x1 and the x2 are, which is the level of expression for gene 1 and gene 2. And what I would find is that the mean expression was this. That shouldn't be surprising. It's sort of midway between 1 and 50 for both genes. And then I would find the eigenvalues and eigenvectors of this covariance matrix. And that's the more interesting part. What we learn from that is that there's a new coordinate system. And then that new coordinate system is what I'm calling G1 new and G2 new. And now what I find exciting about it is that, and note also the eigenvalues. The eigenvalue is huge for the one direction, and it's very small for the two direction. Well, what I carry away from that is a suggestion. And the suggestion is, hey, build your theory, your reduced theory, around strictly the G1 degree of freedom. The G1 new degree of freedom is the way I should have said it. So, the words that sometimes people use is what fraction of the variance is explained by a particular principal component. I find that not so much to my taste as the way I said the words, which is I see this complicated world. I'm in this room. I'm breathing air. There's molecules. There's 10 to the 23rd of them. There's 10 in this room. There's probably 10 to the 24th or 25th. And I don't want to talk about all those molecules. So I need to find some new degrees of freedom to build my theoretical structure around, which in the case of the molecules in the room is words like pressure and volume and temperature. And here, in this case, what the hope is that by virtue of doing this kind of principal component analysis, we will learn what the relevant degrees of freedom are, and then we can from that proceed. This is kind of cool because it's essentially quantifying how much error you get by ignoring this degree of freedom. I'm not going to belabor it. I'm just pointing it out. This example is one that our listeners could undertake for their own amusement. Now I've got 3 different genes, and those 3 genes live somewhere in this 3 space, and there's a plane, which is in a certain sense the best plane that fits that data, and our eigenvectors, tell us how to describe the positions of those 3 bumps, the red, the green, and the yellow bumps in the plane, and ignoring the perpendicular direction. So once again, it gives us a thought about how we might go about dimensional reduction. The last thing I wanted to say about this particular example, and by the way, I'm a little worried that there's an error right here in the arrow and the repression sign. So that's just to alert people. Here, what I'm showing you is a richer example. And this is an example that also I've asked my class to consider, which is now I have these 3 genes, and what they do is they produce a red protein, a green protein, and a yellow protein. And the red and the yellow and the green protein go elsewhere, and they regulate other genes. And so what that means is that these genes are followers. They follow the lead provided by these guys. And you actually see that because what we note down below is that these bands correspond to what's happening with these genes over here. In other words, I look at the low or high character of red, green, and yellow, and that will tell me about the high or low character of these guys, which are either activated or repressed by red, green, and yellow. So this is a transcriptome, a synthetic transcriptome that's no longer based on the constitutive model, but instead based on solving the differential equations. Just to give you a sense of it. So let's say, dgn by dt might be written as minus gamma gn, and then plus some rate constant, and then 1 over 1 plus green over some k squared. This means that our gene gn is repressed by green. How do I know that? Because as green goes up, the rate of production, which is this thing, of gn goes down. The larger green is, the smaller this thing is, and hence the less production. So it's repressed. If I wanted gn to be activated, let's say by red, then I would have dgn by dt is minus gamma gn, and then plus r, let's say red over some k squared over 1 plus red over k squared. So note what this one does. This guy is an activation function. It's saying that as red gets more and more strongly expressed, this thing is going to go to 1. So I'm going to increase expression. Why am I telling you all this? I'm just trying to point out that one can, in the confines of one's own mind, dream up genetic circuits, networks, and then use the tools that we've talked about in this course to write down differential equations. We can solve them in steady state, for example, and obtain a mRNA count for each and every gene, which is what we did here. And you can go through the same procedure, principal component analysis. This one's interesting because you see that the first few eigenvalues are actually very large. This is a log scale. And then all the other eigenvalues are a lot smaller, and that's telling us once again that we have a dimensional reduction, that somehow the whole story resides in the action of these guys. So that's the kind of thing that's going on. And then my last example is this one, which is these three are merrily going about their business, and then I have tons of other genes, and the behavior of those other genes is based on coin flips. And what we find is all those other genes, they are doing their thing, and when I evaluate the principal components, I will see no evidence of what's going on with these three, because there is no covariance, if you see. In other words, when I construct the covariance matrix, these genes do not know about those genes, and vice versa. So they are independent in the sense that they're going to be separate blocks of the covariance matrix. So I think this is what I had to say for this particular vignette. Just to summarize where we are, this is the almost last thing that I'm planning on saying about representations, vectors, and collective coordinates. And our last example is going to be the example of the very beautiful and interesting example, I want to say, of the motion of C. elegans. So this is really cool. I'm going to refer to it as the eigenworm, and I think that may be what the original people that worked on this called it. I don't remember anymore, but we're really going to, so it's a very different situation. Earlier we did normal modes of a mass spring system. Now we've turned to transcriptomes. Now we're going to turn to motility of multicellular organisms. Two or three quite distinct examples.